Okay. Hello, everyone. Welcome to this live session, and thank you for joining us, at least on this sunny day in Manchester. It's bright skies today, and it's lovely to see you all here with us this evening. So, as you know, this month is our cancer-themed month, and this is our second live session of the month, which is going to be a live talk with Dr. Isabel Pires from University of Hull. So, Isabel, sort of her research focuses on, like I say, on cancer, and she specializes in understanding how cancer cells utilize oxygen to help themselves. So before starting to learn about this project, I would like to introduce our structure to those who are new to our platform. So in the next half an hour or so, Isabel will be Isabel will be telling us about her research, and then we will have a 10-minute comfort break and resume back for a question and answer session afterwards. So during her talk or during the break, if you feel any questions, if you have any questions, please feel free to post your questions on our social media accounts, email, or on YouTube channel using the chat function on the right-hand side. So during the break, I will collate these questions and will be asking these on your behalf once we resume back for the quick Q&A session. And during the Q&A session, if you feel like you want to follow up a question, again, you can submit your questions and I will be checking the questions constantly from the YouTube channel. So different than our structure in live talks, today Isabel has actually planned an interactive session in which you can join in via a quiz. So what you will need is to grab your phones or any other media or any other device that you can connect to internet with and go on to mentimeter.com. So Isabel will be providing you some codes and you will be entering those codes into this website and you will be then able to answer the questions asked. So I'm afraid this time you will have to work as hard as our guests. So before moving on with our session today, I would like to briefly introduce Isabel. So she is a colleague of mine, first of all, and it was a pleasure to meet her years back. She's a cancer biologist, and as I mentioned, she's a lecturer in biomedical sciences at the University of Hull. She has a particular interest in the biology of low oxygen in tumours and their effect and how they affect treatments and diagnosis. And she is a very active public engagement person who actually has been featured in a TED Talks a couple of years back. So Isabel, thank you very much for being with us today. It's an absolute pleasure to have you with us, and I look forward to listening to your talk.
Thank you so much for the invite. So, as usual, is sharing the screens, and hopefully everything's gone to plan. So hopefully you can all see my screen. So thank you so much for the invite. It's a pleasure. And actually, I've got a link with Manchester because I did my PhD in Manchester and lived there for 6.5 years. So it's a place which is very, very close to my heart. So it's really lovely to be speaking via this Manchester organized event. So as was mentioned, so my main research into, so I'm a cancer biologist. I'm what's called a basic biologist. So I'm interested in biology nitty-gritty. And mostly at the moment, I'm really interested in how low oxygen conditions can affect biology in the context of cancer. So what I'm going to be talking to you about today is some key concepts about low oxygen biology, what we know, why do we care, and how do we deal with it. So as it was mentioned, we are going to be doing some quizzes. So this is something I use with my students. Which is Mentimeter. So if you go to the website, www.menti.com is at the top of the screen, so you should be able to see it. And then you type in this code up here. So 8183372, and just tell me how you are today. So just 1 or 2 words, and it will appear as a word cloud. So I'll give it like a minute or so we can gauge how people are today. And I guess it's to gauge to connect with the technology. Oh, hello. Yes, good. I've been marking all day, so I'm a bit tired. It's that time of year for lecturers. Yeah, see if a couple more people just so that we are seeing who's there. And there's a bit of probably a lag of getting the code. And again, don't feel like you have to interact. This is just a way of kind of, because normally when I give a lecture like this, I ask questions to the public, and obviously I can't. So this is my way of kind of interacting with you a little bit. Oh, happy, that's a great thing. It's, as I was mentioning, that it's sunny in Manchester. Sunny in Manchester, it's really a rare occurrence. And Hull has been sunny, but a bit rainy as well, which we get less rain, definitely less rain than in Manchester. Right, well, thank you, everyone. Again, people can pop in and out, so don't worry, you don't have to engage with everything, but thank you so much for engaging. Right, so the talk is going to be about low oxygen conditions. So why do we care about oxygen? So again, you should be on your phone again. Why do you think we care about oxygen? You can type in full sentences here. So why do we care? Why do we care about this particular molecule? Because it's amazing. It is quite a cool molecule. It's quite interesting molecule, very reactive. So why do we care? Anybody has any other ideas? The odd thing with this, and it's been the kind of the thing about virtual lecturing, is that you don't get the immediate response and see how people are looking. Probably important, yes, it's quite important in biology. Fundamental to life as we know it, and the as we know it is quite important. For most life, there are some organisms who don't need it. It keeps us alive, keep us alive, keep us alive, metabolism. Brilliant, yes, so metabolism brings me very nicely to my next slide, which is we care about oxygen because oxygen is required for our cells to be able to take sugar, break it down, and get energy out of it. So without oxygen, well, we can, this is a process which are oxygen dependent, but that's kind of the main way. And this happens in what people always call the powerhouse of the cell, the mitochondria. And it happens on the inside of the mitochondria through a process called oxidative phosphorylation. And oxygen is one of the key components of that process to allow the changes in electron potential that lead to the production of oxygen. Sorry, the production of energy. So one thing that is really important to establish here. So when we're talking about normal oxygen levels, and you probably have heard, unfortunately, through the kind of the news last year with COVID about hypoxia and low oxygen and all these sort of things. So what do we consider? So normal atmospheric oxygen conditions and physiological oxygen conditions, because they're very different things. So in the atmosphere, oxygen condition, oxygen tensions, and I'm expressing these here as percentage of oxygen, because that's an easy way to understand. There's other measurements for it, but it's a good way to understand. In terms of percentage of gases, atmospheric oxygen is about 20 to 21%. Anywhere below 20%, 19% or lower, it will really affect a human body. But when we inhale air, once it passes through the alveoli in the lung and into the arterial blood, it actually drops quite a lot in terms of the percentage in the blood, and it's actually about 12% oxygen in the blood. And when you get to the tissues, so the peripheral tissues, it actually drops in. Is very, very depending on which parts of the body. So another thing to remember is that it depends on which part of the body we're talking about. Things like the eyes can be quite low oxygen, so even 1%. The heart, the kidney, and the liver, again, vary a lot. Some parts are more oxygenated than others. And the uterus, again, is quite a low oxygen part of the body, which again, 3 to 5%. And some of these are relevant because of some of the biology I was telling you about. So as I mentioned, so we got 21% oxygen in the air, we inhale it. So again, it's important to understand how we get oxygen from the air to our cells. So through ventilation, we inhale, then there's pulmonary gas exchange at the capillaries in the alveoli. Then the oxygen is captured by the red blood cells, is carried around the circulatory system, and then diffuses at the capillary beds in the tissues, and it gets into the cells where it can be used by the mitochondria. So guys, it's important to understand this kind of process. And I will get to cancer at one point. Don't worry, this is not just about human physiology. So with all this in mind, so we probably again heard about hypoxia in the context of COVID, and hypoxia is low oxygen, but is low oxygen where? So what is hypoxia? And again, another Menti quiz, and this time is a multiple choice, so you get to try that as well. So is it low oxygen content in the air, low oxygen content in the blood, low oxygen content in the tissue, or low oxygen content in the tumors. So which is hypoxia in terms of biology? I think I should give you a minute or so to have a think. Anyone? So in the air, in the blood, in the tissue, or in tumors? So got 1 vote for blood, okay. 2 votes for blood, 1 vote for tissue. Anyone else want to have a punt? So the person who said low oxygen tissue is correct. It's low oxygen in tissue. Now, actually, it's also low oxygen in tumors because tumors are composed of tissue, but by definition, hypoxia is considered an inadequate oxygen perfusion of a tissue. So when you have lower than normal oxygen tensions, this is what we call percentage of oxygen tension within the tissue. And again, this is why it's important to remember that range, because it depends on which tissue you're talking about, what it'd be below normal range. So normally, the broad definition of hypoxia, if you consider like the average probably of oxygenation of tissue, it can be considered between 5% all the way down to 0. And this is 5, it could be 3%, again, it depends on the tissue as well. And this is to do with the little molecule, which I'll tell you about later. So the 0% is actually has a special name. It's called anoxia rather than hypoxia. Hypoxia literally means hypooxygen, so low oxygen. And anoxia is absence of oxygen, so no oxygen. And in fact, you might think, well, Isabel, is there anywhere in the body which is anoxic? Well, there's some areas which get close to that. And in tumors, you actually see regions of anoxia. So it is biologically relevant. Now, it's really important not confuse hypoxia in a tissue with hypoxemia. So hypoxemia is when your blood is not carrying enough oxygen. So this could be, for example, due to something like an anemia, or due to, for example, pneumonia or COVID. You can get hypoxemia, which is this low contents of oxygen in the blood, which then can't diffuse into the rest of the tissue. So it can drive hypoxia in the tissues, but itself is not hypoxia specifically. Now, in cancer, we get to cancer finally, hypoxia is derived from very specific characteristics of tumors. So tumors grow really fast. They are very oxygen greedy because they're growing so fast. And they also, because they grow fast, they grow away from the normal blood vessels of the tissue where they originate, which means that the oxygen can't get into the tissue area. So oxygen actually has a very small diffusion range. It's about 0.15 millimeters, so 150 micrometers. So below that, you start seeing the oxygen level starting to drop. So in tumors, an amalgamation of rapid tumor growth, so tumors growing fast and out of control, high oxygen demand because of that, but also this irregular vascularization, which means is like blood vessels are irregular, and I'll show you some pictures of that in a minute, and the inadequate perfusion of the tumor mass. That's why you end up with regions of hypoxia in tumors. And this is something that we know occurs in majority of solid tumors. Now, the other thing that you need to know about hypoxia is that hypoxic conditions in tissues are a stress response, cause a stress response. So hypoxia per se is not nice. Our bodies don't, or the body don't, tissue don't like to be in low oxygen because they need the oxygen, right? So very rapidly, it triggers an adaptive response to try and get the oxygen levels back to normal, or to try and survive long enough until you get the oxygen levels back to normal. And this happens in a lot of conditions. So in normal physiology, so during development, in utero, actually the embryo is subjected to low oxygen conditions. If you remember, I mentioned the utero has 3 to 5% oxygen itself. And actually, we know that hypoxia is important in utero for normal development, things like vasculature in the circulatory system of the embryo, the heart, the spinal cord, and a lot of actually requires that in the embryonic stage, that low oxygen level is important. However, normally, so for example, if sometimes it happens that baby could, during birth, be trapped, or if the mother smokes, or if there's issues with oxygen below that hypoxia, normal hypoxia for the fetus, that could lead to developmental issues. Also during exercise, so if you're running and you start hyperventilating because you're running your muscles. The reason why you hyperventilate is because your muscles are eating so much oxygen that they basically trigger a response to tell your brain, hyperventilate, make the heart pump faster so the tissues can get more oxygen. Also, during altitude adaptation and altitude sickness, oxygen level hypoxia plays a big role in this. But actually in pathology, so during disease, hypoxia can also play a big role. Now, today I'll talk about cancer, but it's important to note that other diseases also have a big component of this hypoxic response. So things like ischemic disease, so strokes, heart attacks, in which is blockages of blood vessels lead to that parts of the brain or the heart become hypoxic, and that can again trigger those survival processes. And lung disease, so again, COVID, I promise I'll stop mentioning it, and things like pulmonary hypertension, that can originate hypoxia within those tissues as well. But we're talking about cancer today, so we're going to focus on what is tumor hypoxia or hypoxia in cancers and what's its role. Now, to be able to understand that, we need to understand how does hypoxia causes this adaptive response. So there's two processes, broadly speaking, by which this happens. There's a neuronal part and a transcriptional part. And what I mean by transcriptional part is changes in expression of genes to change the biology of the cells. So in the neuronal part, that's due to the role of a small mass called the carotid, carotid body, which is just above the carotid artery, and it communicates directly to the brain. Is this chemosensitive cells that can tell if the oxygen content of the blood is lower than it should be, and it tells the brain, okay, we need more oxygen in the blood. And that will lead to things like, for example, during exercise, hyperventilating, so increasing cardiac output, and getting just the blood, air to come into the lungs more frequently, and for that oxygenated blood circulate through. So that's a neuronal regulation. But actually, a really important part of this as well is this change in biology, this transcriptional change. But we mean transcription, like I said, is changes in gene expression, which will change the biology of the cells. And this is what happens to allow the cells to survive in these pathological conditions or to adapt, for example, doing altitude adaptation, to these conditions of low oxygen that are not normal for the cells and are prolonged. So this happens mostly through a very, very important protein, or actually two proteins called the HIF family. So HIF stands for Hypoxia-inducible factor, and it's actually a pair of proteins that interact with each other, and they can bind DNA and change expression genes. The interesting thing to know about this is that there's two subunits, alpha and beta. And again, without going to a lot of the biological detail, and again, if you're interested, ask me about it afterwards. The alpha subunit, its activity and stability, so it being present, is dependent on oxygen. The beta subunit isn't. So it's this alpha subunit specificity and dependency on oxygen, which makes it oxygen responsive. So what happens normally is that in normal oxygen conditions, normoxia, we'll call normal oxygen conditions, the alpha subunit is not present. So it's constantly being degraded, thrown away into the cell rubbish bin, and it's not active. But very quickly, when cells experience conditions of low oxygen, it can rapidly, what we call, stabilize. So something happens that it makes it stop being destroyed all the time, and it can then increase the levels very quickly. It can bind to the beta subunit, can then bind to DNA, and then transcribe the genes. And these gene expression changes can happen in all sorts of ways. So a key one, and one of the first ones to be identified, is actually changes in a gene called EPO, which codes for the protein erythropoietin, which is very important for making more red blood cells. So again, seeing how adapting to low oxygen to increase the capacity of the blood to carry oxygen. It can also drive things like changes in metabolism. It can drive things like recruiting blood vessels to try and get more oxygen in. It can make the cells survive for longer, etc. And again, we'll cover some of those a bit later. So that's HIF, and HIF is probably one of the key proteins in hypoxic biology. There's others, but HIF is kind of the cornerstone of it. And it's so important, in fact, that understanding how HIF functions and how oxygen levels are sensed and responded to was actually awarded a Nobel Prize in 2019. So all of us in the hypoxia field were like, yay, finally! And it was awarded to these three men. So Bill Kalin, who's based in the States, Peter Radcliffe, who's based in Oxford, and Greg Menzer, who's also based in the States. And between them, they understood the various parts of the mechanics of how the cells respond to oxygen, so the role of HIF, how HIF is regulated, how it works, what's the other, because it's quite complex, as you can imagine, it's quite a complex process. So they were key fundamental in understanding all this. But the thing about Nobel Prizes, they tend to be dominated by men. And it can get a bit frustrating. I'm a big, as part of my public engagement stuff, I do a lot of activities. I was a speaker for Soapbox Science, which is a platform for increasing the visibility of women in science. So this is exactly what I'm going to do now. And before the Nobel Prize and before hypoxia was even a thing, and they knew about HIF, Mabel Purifoy Fitzgerald was a British physiologist and clinical pathologist who was fundamental to understand, whose work was fundamental to understand that that humans could adapt to low oxygen conditions, so altitude adaptation, and also the role of hemoglobin in this. So although she wasn't really, at the time, they didn't have the tools to understand about these molecular processes. At that stage, we didn't have the tools, but she was fundamental to understand that. Mabel was quite an impressive woman. She was the first female student in the medical school at the University of Oxford in 1887. She wasn't awarded a degree. However, when she was 100, she got given an honorary degree and an apology by the Oxford Chancellor, Vice Chancellor, and also became a member of the Physiological Society. And again, just highlighting some of the hidden women in the science of hypoxia. To be fair, she was mentioned in a Nobel Prize speech by Peter Radcliffe. So again, credit where credit is due. So this hopefully highlights how important hypoxic biology is. So as I mentioned, in tumors, hypoxia is caused because tumors grow fast, they need a level of oxygen, and the vasculature is not working very well. And actually, we know it doesn't work very well because we can take casts of vasculature in tissues. And what you can see here on the right, on the top is normal tissue, and on the bottom is the equivalent cancer tissue. So normal skin, melanoma, and normal skeletal muscle, and sarcoma, which is a type of muscle cancer. And hopefully these are casts of the blood vessels for those tissues. And hopefully what you can see is that in the normal skin, they are well organized, they're well distributed. But when you compare it to the melanoma, they're all over the place. They're really branched out, they're really irregular, they're not really that well organized. And there's areas which aren't covered by vasculature. More even starkly different is the vasculature in skeletal muscle versus the tumor equivalent. So you can see skeletal muscle vasculature, very regular, very well organized. All the blood vessels look like the same way, all very long and nice. Look at the sarcoma. They have this characteristic, what we call serpentine, because it's this wiggly, looks a bit like the Amazon or like a snake. It abruptly ends, it's got branched out, and they're also, at a cellular level, very leaky. So as well as being irregular, they're very non-productive, which means that although the tumors are trying to make new blood vessels, that actually don't work very well, which can make the oxygen conditions within a tumor change quite a lot. So one thing that I always remember about cancer is that tumors are very dynamic entities. So they change a lot, they're very diverse genetically, but it also, oxygen tensions can change a lot. So because of this revascularization, you got these cycles of hypoxia and reoxygenation. So oxygen tension within a tumor can be chronic, which is there for a long period of time, but they can change rapidly, sometimes within 20 minutes or less. And this can be crucial for the cells to become even more aggressive and becoming more tumor-like and more likely to spread. So as well as having these changes in time, we also have in tumors different oxygen tensions. As I mentioned, normally in hypoxia, we consider about 5% to 0. So in tumors, we normally say that if it's like at 1%, 2%, we call that moderate hypoxia. Below 0.5 towards anoxia, we call that severe. So that, because there's slightly different biology associated with each of those. And this is just a little diagram just showing again the very strange nature of the tumor vasculature with these blocks, these leaky vessels. And some of these occlusions can be just the anatomy. It's just the growth, the mass that grows, it compresses the blood vessel and then it can release it. could just be the anatomy as well. It's not just the blood vessels themselves. So one thing before we go next in terms of, so actually, what are all these biological changes you keep talking about, Isabel? I mean, we're having some cancer lectures, but just to highlight that one thing that's really important to understand about cancer is that there's key biological characteristics, which we call the hallmarks of cancer, which make a tumor. And this can be things like there's changed cell and metabolism. They can keep on dividing as long as they're given enough space and food, that the growth signals are on, the non-growth signals are off, immune system is changed, they're very genetically unstable. And again, there's something which is a bit complex, but that means that they're very likely to acquire mutations, and that can make them a bit more aggressive. The cells don't die very much, so this is escape to cell death. Like I said, this revascularization, which we call angiogenesis, happens all in tumors. It's a very highly inflamed environment because of all these cytokines and all these really leaky vasculature and all these things happening. It's quite an inflamed environment, but also very crucially that tumors to be tumors have to have the ability to spread elsewhere in the body, because that's unfortunately what kills most people, is this ability of tumors to spread. 90% of deaths associated with cancer are the cells that spread elsewhere, to the bone, the brain, the liver, the lung, for example. So I just wanted to mention this because when we talk about how hypoxia changes biology, a lot of these are affected by it. So most of those hallmarks that I mentioned are affected by tumor hypoxia. The one particularly on my work, we're particularly interested in a couple of these. I'm interested in this change in genomic stability, so this ability for the cells to acquire more mutations, because we know there's some biology that changes that can drive the changes that lead to this increased genomic instability, increased mutation gain. But we also know that we have this increased likeliness to spread, also driven by low oxygen conditions. So a lot of the key HIF gene targets are driving this biology. But one thing that's very unique to the hypoxic context, there's other microenvironmental, so tumor characteristics that do it, but particularly with hypoxic context, is that it's associated, as well with all these changes in biology, is associated with resistance to chemotherapy and radiotherapy, which is an enormous problem. So as well as changing metabolism, which makes sense, you don't have as much oxygen, so you have to change it to an oxygen-independent and aerobic biology. You switch angiogenesis on because you want to recruit those blood vessels, et cetera, but also you drive this resistance to chemo radiotherapy. And particularly with radiotherapy, which is something we work on in my lab, this is purely due to the fact that radiotherapy to work properly needs oxygen, because it needs it for the damage to take place, to be set in place, and the cells to die. Without oxygen, you can see that radiotherapy resistance is a big problem, and chemotherapy drugs that depend on that as well. So that brings me to, why do we care about tumor hypoxia? It might just be, I mean, I'm a biologist, I'm interested in this, how this low oxygen conditions drive all these changes. I mean, that's something I find fascinating. But why do we care? Now, we care because high hypoxic content, which means when tumors have a lot of low oxygen regions, this is associated with patients doing worse. So we know that both in terms of, these are Kaplan, we'll call Kaplan-Meier plots. So these are plots, survival plots. And what you need to note here is that if the curve is lower, that means these patients are doing worse, because this is a probability of survival. So if it drops, unfortunately, that meant that patients were lost. So the higher the curve, the better, and the lower the curve, the worse. And for both of these, this is just two different tumor types, cervical cancer and head and neck, but there's other ones done, showed very clearly that patients that have a higher hypoxic content, which is the patients on the full line here and here, do much worse than patients that have a lower hypoxic content. So have better oxygenated tumors, which kind of doesn't make sense. You would think that they will grow faster, but actually all these things that hypoxia can drive means that they have poor prognosis. They're likely to do worse. And this is not just in terms of survival, but in terms of likeliness to having this disease spread, and also the likeliness of responding well to therapy as well. So this is why we care. This is why understanding hypoxia is really important. So just to wrap up, so I've hopefully got a gist of why hypoxia biology is important, what it is, how it's regulated. So what can we do to understand and to tackle tumor hypoxia. So the first thing we need to do is study it. So in the lab, and because I'm, like I said, I'm a basic biologist, I mostly do work in the lab. So I don't do much work with patient samples. So there's several strategies we can use to mimic hypoxia in the lab. So one of them is by actually using drugs. Weirdly enough, you wouldn't think that some drugs could mimic low oxygen, but they can, because there are drugs that basically can basically work as if oxygen isn't present. This is what we call a hypoxemimetic. And what they will do is they specifically will block the degradation of HIF alpha and make it accumulate, which then means that cells will have the HIF response in the presence of oxygen. So they are very useful. They're a bit crude because, as I mentioned, hypoxia is not just HIF, and there's a lot more complexities to it, but it's a kind of a good initial tool to look at. Other things we can use is, so in the lab, my lab uses a lot of cancer cell models. Normally we grow them in Petri dishes or in plates, but actually you can also grow them in 3D structures, and this is called spheroids, and they can be more or less complex. But the lovely thing about spheroids as a model is that because they are 3D spheres, and you can grow them into quite chunky, you can get like 1 millimeter big spheroids. That means that we can mimic the oxygen diffusion gradient from, hopefully you can see it here. In the blue is the more oxygenated and red less oxygenated, going from the outside of the spheroid to inside. If you remember, the oxygen diffusion range is about 150 micrometers. So a spheroid which is less than 0.5 mil will have hypoxia regions within it. And we can do all sorts of things with them. We can measure them. We can stain them for hypoxic markers. So this is a well-known gene that is a HIF target. And we can use dyes that specifically accumulate in hypoxic regions that we can then look at under the microscope as well, which again is a synergy drug. It's a quirk of its chemistry that in low oxygen conditions, it changes, it can't leave the cells. But my favorite way, although spheroids are very close second, but my favorite way of modeling hypoxia is using hypoxia chambers. So this is my own hypoxia chamber in my lab, and that's actually me many years ago when I had short hair. What it is basically a massive incubator, analogous to if you ever watch The Simpsons and you see Homer in the beginning, kind of radioactivity, and he's kind of handling radioactivity inside a box. It's pretty much that, but not for radioactivity, but for low oxygen. So what we have is our cylinders of gas mixtures, and we pump in nitrogen, air, and CO2, so we can then adjust it to make it more or less hypoxic. And then we just grow the cells. As you see here, I'm holding a dish of cells in there, and we can do all sorts of things in there. We can image them, we can manipulate them, we can harvest them, and then we can reoxygenate them, bring them out, put them back in. It's great because it's sealed, it's got an interlock system, and it's got these very cool gloves you can use to go in and manipulate the cells. It looks great. People love seeing people handling it, but actually it's very boring to use and quite sweaty because it's 37 degrees inside, so it's quite warm. So it's quite a cool methodology to use. Now, this is how we study hypoxia, and there's other methods as well, and I just don't have time to cover them all today. But obviously, as I mentioned at the start, I'm very interested in targeting hypoxia, exploiting it therapeutically. So what we want to do is target hypoxia to try and make cells respond better to drugs or to make them more sensitive in general. So what we're doing is targeting using HIF. Now, HIF targeting has been challenging, hasn't really worked because it's such a crucial molecule, it's very hard. But a recent drug, and I can go into more details later on, is actually showing some promise in renal cancer, which is kind of very heavy HIF biology in there. So we're very excited about that. And also, there's a lot more biology to hypoxia than just HIF, so there's a lot of targets. And the more targets you have, the more you can work on. So in particular, I've been working on this bit down here, which is targeting very particular quirk of severe hypoxia, which again, ask me about it at the end. All right, so that's my overview, and hopefully you understand that from understanding how it behaves to targeting it, we are making strides about using hypoxia against the tumor. And actually the flip side is that in cardiovascular disease, you can actually do the reverse. So again, ask me more about it later. So before we close, I just have a very short quiz. So if people get into the quiz, you just click on there's like a little cat ears at the corner. Normally it's a little heart, but I thought there are cat ears in there. Just to test your hypoxia knowledge. So I'll just bring in some people just to click on. Hopefully you're still with me. I haven't lost all of you yet. And it's only 5 questions. It's very short, and they're multiple choice. So, But the thing about this quiz is that it's timed and you get points. So we might get a big competition going on. So my students love this because it's a revision and it's competitive. So, okay, there's a few of you going. So in the next page, you have to give yourself a name. You'll get a little cute icon. You unfortunately get randomly allocated icons. There's some really adorable ones. And when you have a class of like 40 students, you get tons of these and they're very cute. Right, let's start the countdown. So good luck, as I said. The faster you are, the more points you get if you get it right. So it's a trade-off there. Right. First question, it's about 20 seconds. Which of the oxygen levels below are hypoxic? 21%, 12%, or 1%? Need a countdown sound. And the answer is 1%. Next. Again, they're all the same. They're all fast. It just does that to remind people. So what does HIF stand for? Is it hypoxia influence factor, hypoxia inducible factor, hypoxia invisible factor, or hypoxia is fantastic? It's hypoxia inducible factor. You're all paying attention. Right, next. Sorry. Which of these is a driver of Hypoxian cancer? So, is it decreased tumor growth, resistant to radiotherapy, irregular vasculature, or all of the above? So which one of these makes hypoxia occur in tumors, from what I explained before? It's a bit difficult. It's a bit more difficult one. Oh, everybody all got it right. It is irregular vasculature. All doing well so far. Next one. Which of these is linked to tumor hypoxia mediated poor prognosis? So which of them are characteristics of hypoxia that mean that patients do worse? And instability is genomic instability, by the way. I don't know why that is deleted. It's all of the above. Well done, people that picked it. So increased genomic instability and increased angiogenesis are also drivers. And last but not least, last question. What is PT2385? This is did you spot the drug name? Is it a hypoxemimetic, a HIF target gene, a hypoxia PEG tracer, or a HIF inhibitor? Ooh, this is paying attention to the slides. Oh, this is a tricky one. It's an inhibitor. So that person, if you got all of them right, you might be the winner. Let's see. Leaderboard. So Homer, I like that. Homer won. Congratulations to Homer. And well done to S, Earthman, and DB as well. Well done. Thank you for playing. And are there any questions? And obviously, pass the questions, we'll leave them there. And hopefully you've enjoyed the talk.
Lovely. Thanks very much, Isabel. It was a very interesting talk. So we're just going to give you a break now, considering time. So shall we do a 5 to 6 minute break and resume back at... Okay. Lovely. Thanks very much for holding on with us while we're having the break, and thanks very much for submitting the questions. So I'm going to start with one of the audience questions. So Isabel, are there any genetic changes that can predispose someone to hypoxia-related cancer problems?
There are some syndromes which can lead to HIF issues with like HIF being abnormally high. So there's one called von Hippel-Lindau syndrome, which is actually one of the it's called this, and actually it's a molecule which is important for, it's the one that targets HIF for degradation that makes it be degraded. And in particular, some syndromes, and it seems to be things like renal cancers and some central nervous system cancers, which have this defect. So that's one that definitely would predispose. They're very rare, as many of these syndromes that we learn from are. But yeah, there's some cases. But in general, just fast-growing tumors will tend to have more hypoxic regions. But there's definitely syndromes that can lead to that.
Okay, great. In addition to this, are there any drugs yet in practice that specifically target hypoxic tumors?
No, I wish. There's a couple. This one that I mentioned, the PT2835. So this is actually in clinical trials at the moment. It's showing a lot of promise. But it's been really hard because the issue with drugs that target HIF biology is that HIF relies on protein-protein interactions, which are not super strong. So it's not an enzyme. Targeting enzymes is much easier. So because they normally have activity pockets, you can build something that blocks it. In the case of HIF, you're basically trying to block either the two subunits binding or it binding to DNA or something binding to it. So it's much harder to target. But like I say, there's promise with those. What there are drugs that exploit the hypoxic biology or hypoxia-adjacent biology. So there's drugs which are in the clinic which target angiogenesis, for example, which is this blood vessel sprouting, which is not just associated with hypoxia, but is very linked with it. And this is through drugs like Avastin, which is bevacizumab, which basically blocks the signals that makes the cells sprout the vasculature.
Okay, great, thank you. So what do you think is holding back drug development for hypoxia applications?
Bad clinical trial design, in my opinion. Big issue we have, and it's not bad clinical trial design, I'm being a bit facetious, it's that we don't have still very highly accessible tools for determining how much oxygen, what's the hypoxic content in a tumor. So the blots that I've shown you, they're done using a thing called an oxygen electron probe, electrode probe. So it's basically like a needle type probe that you insert into the tumor to measure the oxygen. As you can imagine, very invasive, need to have an accessible tumor that's big enough. So that's why cervical, head and neck, prostate, breast tend to be the ones that we have that data from. There are molecular imaging techniques, so like PET, which can be used for giving a gauge of hypoxic content. So there's the most commonly used PET tracer, F18FTG, is basically a glucose analog, and it accumulates in these regions that are very metabolically greedy, but not all of them will be hypoxic. So it's not a great biological marker, but there's actually a lot of studies looking at improved PET, which is much more hypoxic specific. So actually, in my lab, I've got a collaborator and we're working on that at the moment. But it's not just us, there's a lot of people throughout the field doing that. The other thing that we can also do is to take biopsies and look at gene expression signatures, what we call hypoxic metagenes, which are representative of a hypoxic response being present in the tumor. But again, because it depends where you take the biopsy, it's not going to represent the whole tumor. So I think that's what's been holding it back, is when we have better idea of exactly what the hypoxic content is, we can design clinical trials that will actually be used for patients with high hypoxic content. The issue we've had, there's been a lot of really cool, promising drugs that failed at the clinical trial stage. And we think that the reason that happens is because you have a lot of patients that have hypoxic content and some that don't, and that dilutes down the data. So better imaging, hypoxic diagnostics will probably lead to more drugs coming through.
Okay. Lovely, thank you.
Yeah.
And do you see those kind of drugs being used on their own or in combination with other things? Do you see them being used as chemotherapy agents or just prevention agents?
I'm a big fan of combination therapies. I think for hypoxia, you want to do it in combination, because you want to do is target the biology and then whack it with something that you know works. Because if you got something like radiotherapy, one of the biggest tools for treating cancer. So think surgery first, radiotherapy, chemo, and then specific expensive immunotherapies coming on the bottom. They're cool, they're very effective, they're very expensive, very hard to deliver. Combining something which has a more kind of whack approach, like something like radiotherapy or conventional chemo, with a more signaling specific, means that you are exploiting that synergy of the signaling with the approach. And if you have tumors that are very sensitive, radio resistance, and then you sensitize the hypoxic regions, you can make them more radiosensitive. So, but yeah, good question.
Okay, lovely, thank you. So, all right, so somebody's mentioning that All right. So you have mentioned in your talk that there are complex mechanisms involved in hypoxia, which includes molecules other than HIF.
Yes.
So, and you mentioned that you can target these as well for therapeutics. So could you give us an example of that?
Right. Actually, something I've worked on in the past. So I'm quite excited. So before being a lecturer at Hull, I was a postdoc at Oxford University, and I worked in the lab of Professor Esther Hammond. And Esther's work was primarily looking at radiosensitization, but particularly looking at the biology of anoxia. So not even like what we call radiobiological hypoxia, the one that drives the really severe radioresistance. And one thing that happens in those conditions is that we see that there's a lot of signaling pathways that get turned on, which are similar to when you have damage to DNA, but without damage to DNA. And the cool thing about these signaling pathways is that we have drugs against them, or they're kinases, so they're enzymes which are very druggable. So one of the things that I did when I was in Oxford was that I worked on a particular molecule, ATR, and it's one of these key signaling molecules in this DNA damage response, because it is that, although there's no DNA damage. And actually, the cool thing about that work is that I did a lot of kind of the initial biology stuff, but now it's actually in clinical application. So it's one of those that has gone to the clinic. Now, it's widely used not just for hypoxia, but actually something that can be used to sensitize hypoxic regions as well. So it's since I was a PhD student, I always worked with small molecule inhibitors, kind of targeting specific pathways and combining them with things like radiotherapy, chemo. So that gets me really excited. And it was just great seeing something that I worked on a prototype molecule of now being clinically available, or being given to patients. It's just
Yeah.
It was great. Exactly. Absolutely great.
Exactly. Lovely. Thank you very much. So there is a comment in the chat as well saying that they love the quiz. So thanks very much for integrating that. Yeah, thumbs up.
No problem.
Definitely.
I'm happy to do that.
Yeah, definitely. Well, in the interest of time, I think we haven't got any other questions, and I'm just going to leave it at that for today. And thanks very much for joining us. And everyone, see you next month with our new theme. I'm sure our volunteers will be posting on our social media and emailing you to let us know about what to expect in June. Take care.
Yeah. Thank you so much, and great questions, by the way.
Thank you.
Thank you.